Hello, good afternoon. As we have already learned about the T cells in the last video, we have seen that T cell brings a specific defense mechanisms. Similarly, today we are going to look at how the B cells get selected and activated to bring about the specific defense in our body. But prior to that, I would like you to again recall some of the things that we have already covered. What we need to know is just as a reminder, we need to know about the ancestors of these different cells like eosinophil, basionophil, neutrophil, macrophages, natural killer cells, T cells and B cells. So what you have to do is you have to trace down its progenitors. So please try to understand this. That's why I just wanting to show you here the different cells, cell lineage. The lymphoid cell lineage is very important because you look at that hematopoietic cells gives lymphoid tissue or the lymphoid stem cells. Then the lymphoblast give rise to the natural killer cells, and then lymphocytes B and T. B cells again further give rise to plasma cells. Alright, I would also like you to please emphasize on this lineage which I just explained to you. Right, so with that, this is just a reminder. Now in the previous slide, I just laid this one of the slides I made incomplete in explanations in the T cells. There are 4 different types of T cells: T cytotoxic cells, T helper cell 1, T helper cell 2, T suppressor cells. These T cells actually helps to the different antigen presenting cells, like T cells act directly on the infected cells and destroy the infected cells. So this brings about cytotoxicity, meaning it destroys. So the cells that is being infected will get destroyed by these T cells. T helper cells act on, also Th1, T helper cell 1 acts on the macrophages and also act on the dendritic cells. They are the antigen presenting cells, so they induces these once these macrophages present the pathogens or the foreign particles to the T helper cell. T helper cell will produce certain chemicals which will signal the other T cells or the B cells or the neighboring cells. So interferon gamma or the TGF beta will help to transmit the message to the other T cells or the neighboring cells. Likewise, T helper cell 2 acts on the B cells. These B cells also treated as antigen presenting cells. B cell accepts the antigens and present to the T helper cells. T helper cells will produce interleukin 4, interleukin 6 and interleukin 10. These chemicals again induces other macrophages and the T cytotoxic cells to destroy the antigens. Therefore, this is T helper cell 2 directly acts with the B cells. Then there is a T suppressor cells, which is also produce TGF beta and interleukin 10, but however these will actually brings the reactions in a reverse manner. This will controls the further reactions to the antigens. One thing that you have to keep in mind is these cytotoxic cells on the surface of the cytotoxic T cells will have protein surface receptor CD8, and macrophages and B cells, T helper cell 1 and T cell helper 2 will have surface receptors CD4. So this is the difference that you have to keep in mind. Cytotoxic T cells has got CD8. T helper cell 1 and T helper cell 2 will have surface receptor proteins CD4. That's all about the T cells. Now we are going to look at the differentiations and activations of B cells. When you look at that, B cell is produced in the bone marrow, as you already know that. Even the T cells also produced in the bone marrow. The B cells, although produced in the bone marrow, it moves to the lymph node and get matured. The prematured B cell move to the bone marrow and is matured at the lymph node. The B cell differentiate into plasma cells and produce antibody specific to the antigens. The third one, B cells also produce memory cells that encounter if attacked by the same antigens in future. So B cell plays a big role in producing antibody and controlling the growth of microorganisms or the proliferations of microorganisms. The antibody acts against the antigens. So what we have to look at is now, what is antibody? Antibody is also known as immunoglobulin. The antibody structure of antibody, you're going to see there are 4 chain of proteins, 2 long chains, inner one are 2 long chain, and short 2 side chains are present in this structure. The first four proteins are the variable regions. 1, 2, 3, 4. These will keep on changing with different antigens. Once the antigen is being recognized, antibody is being produced. Depending on the specificity, these 4 proteins changes. Therefore, this is the region where antigen binds with antibody. These regions is where antigen binds with the antibody, and this region is called variable because it varies from antibody to antibody. The base regions 1, 2, 3, these lower regions are constant regions, and the side chains are shorter chains are light chains, and these are called heavy chains. And these long chain or heavy chains has got FC regions, which is a very constant regions, and this has been these short and heavy chains are being connected by the sulfidal bonds. Now there are basically 5 types of immunoglobulin or antibody produced in our body, and these antibodies are produced for different purposes. And now if you look at how many antibodies are present here, 1, 2, 3, 4, 5 antibodies are present around. Here there is only 1 antibody, there are 2 antibodies and 1 antibodies, and so where there you are going to see that the antibodies are arranged in a ring is called pentamers, and name is immunoglobulin M. Monomer is immunoglobulin G, and this is seratory IgA is a dimer because there are 2 joined base to base. And there is another monomer which is again to have a different function, IgE, and there is IgD monomer. So there are 3 monomers and 1 pentamer and a dimer. So this is just is weight, and then just if in case if you are asked. But what is more important here is what is the function of each one. When the antibodies are produced against any foreign particles, this IgM will be produced. These IgM will be produced to act against any antigens, foreign particles, and this happens only in the initial encounter. When there is a second encounter, there is a second encounter, the plasma cells will produce and subsequently produce antibody, which is IgG. So this is for primary encounter, and this is for secondary encounter. When the second time same pathogens attacks, these will be produced, and then it act against that pathogen. Now serotonin is secreted in the mucous tears and colostrum. So this is for normally in the mother's milk, as well as in our tears, in our saliva. These antibody A are being produced, and therefore it brings a lot of defense in our body. So even our tears contains a lot of antigens A. Antibody A. The antibody of allergic and then parasitic activities are brought over IgE. IgG is all about, because if you have looked at those persons who are suffering from the allergic, these IgE antibodies are being produced, and these IgG antibodies are produced so much, and that causes the trouble to the person who has got allergy. And the last one is IgD. This is found on the B cells. You know that B cells has got surface protein receptors on the membrane, on the surface of the membrane. There are receptors. So on the B cells, these IgD will be acting as a receptors. So this is what you will have to know. There are 5 types of immunoglobulin or antibody, and they have a different functions, and they are found in a different parts of our body. And all this what antibody do, and how they react with the antigen, that we are going to look at in detail later on. So what you have to understand is, here is what is how the structure look like, and how many different types of antibodies are present. Let's continue. In the primary encounter, when the antigens attacks the cell, the antigen presenting cells, or the B cells, let's say here B cells is an antigen presenting cell. So B cell recognizes the antibody, and what it does is this gets activated, and it produce memory cells. It produces the memory cells, and also produce antibodies, but these antibodies are being produced by plasma cell. So 2 things that happens when the B cell get activated: it produce memory cell one, another is plasma cell. These are plasma cells, and these are memory cell. So plasma cells produce a lot of antibodies. These antibodies are IgM, because this is being produced when it has been encountered for the first time. Now these memory cell will be stored for future attack by same germs or the same foreign particles. Now this is a second time encounter. B cell, when there is an infection by the same antigens, the memory cells will immediately recognize the antigens and produce again the memory cells and produce a lot of plasma cells, and then it produces antibody. This antibody is IgG. This is IgG, which I said to you. Initial primary one is IgM, and second is IgG. Alright, this is what is very important. Like these two, although the antibody looks similar, but this is IgM and this is IgG, immunoglobulin G. Now the actions of these antibody: in the initial exposure to the antigen, the B cell produce antibody at a much lower than the second time encounter. So here this is the level of concentration of antibody that is being produced by the B cell. In primary encounter, it produces quite a low concentration of antibody. In the secondary encounter, the memory cells reacts quickly and it produces a lot of antibody. So the response will be much faster in secondary response. Right now, let us go for how the B cell gets activated. As I have already said, B cell has IgD on the surface. IgD antibody. See here, the B cell has got IgD. Now this antibody has antigen binding sites, and that's where here antigen is already being attached to the B cell. Once this B cells gets there, B cells now will be triggered to other cells like T helper cells. These B cells also have a surface receptor protein, which I said earlier. The surface receptor proteins is one is antibody, and there is also MH2 protein, that is major histocompatible complex. So 2 is present, MHC2 are present in all the antigen presenting cells like macrophages, like B cells and dendritic cells. So here B cell has IgD, and once these antigens are presented by these B cells, the T helper cells will recognize the MCH2 and bind with CD4 of T cells. You know that already, I talked about just before. T helper cells has CD4, and these CD4s binds with the MHC2, and these T cells would produce interleukin 2 and interleukin 10, and that activates further other T cells. So this is the activations where you are going to see that cytokinin which is being produced gets activated. Even the B cells get activated, and the other T cells get activated, which will destroy these infected cells later. Right now, humoral response to the T dependent. What I'm saying is how the humoral antibodies are being produced with the help of T cells. So T dependent antigens, like for example, if the bacteria gets infected to the macrophage, you know that macrophage is antigen presenting cell, so it has got on its surface, it will have MHC2 protein. So this MHC2 protein will present the bacteria to the T cells, T helper cells. See, the CD4 has joined with the macrophages and now bound to that, and this is this T cell will produce interleukin, or the cytokinins, or the interleukin 2, interleukin 10, and these will further activate other T cells. Look at here how this interleukin 2 and the other cytokinin is being produced and activate the B cells. Now what happens once the B cell get activated? Once the B cell get activated, it produces a lot of clone cells, clone B memory cells, and clone plasma cells. The memory cells will be retained for the futures in case if there is any encounter by this same antigens, and whereas instantly the plasma cells will produce a lot of antibody, and these antibodies directly move to the bacteria, and it brings about the destruction of that bacteria or the foreign particles. So here antibody acts directly. So what happens is B cell initially activate T cell. T cells produce chemicals that will activate B cells, and B cells produce memory cells as well as plasma cells. Memory cells can be kept for the future encounter. In case any encounter, it will instantly produce an antibody IgD, whereas plasma, it produces instantly here IgM. In the primary encounter, it produces IgM. I'm repeating again and again. Please keep in mind that. So I think with that, this is how the B cells act, produce antibody, and since the B cells is producing antibody, a chemical that can destroy the foreign particle, therefore it is also called humoral defense system. With that, I think I hope you have understood. If there are any doubt, please let me know. You can ask in the class. Alright, with that, thank you.